Welcome back to the deep dive. Our mission today is, well, to dive into something pretty cutting edge in ecology, environmental DNA, or eDNA.
hmm
We know it's this amazing tool for just finding species, especially elusive ones, like a biological smoke detector, maybe?
That's a good way to put it. It really helps find things that are hard to spot otherwise.
But here's the real challenge, the sort of white whale of eDNA work. Can we actually use it to figure out how many individuals are there? To estimate species abundance reliably?
That is the crux of it, isn't it? Moving beyond just presence-absence.
Yeah.
And that's exactly what the paper we're digging into today tackled. It's titled, The Value of Quantitative Environmental DNA Analyses for the Management of Invasive and Endangered Native Fish. And the lead author is Jack Rojahn.
Okay.
Their main goal was to see if quantitative eDNA data, looking at the actual concentration of DNA, could tell us something useful about relative abundance patterns,
Right.
especially, for managing invasive species versus native ones.
Got it. And they tested this in a specific and pretty critical location.
Yes, the Abercrombie River catchment. That's in New South Wales, Australia.
And why there? What's special about the Abercrombie?
It's one of the last strongholds for the native Macquarie perch, Macquaria australasica. It's nationally endangered.
Endangered. Okay. So high stakes.
Very high stakes. And it's facing a major threat from an invasive species, the redfin perch, Perca fluviatilis, a really successful invader in Australia.
Right. So a classic native versus invasive battleground.
Exactly. And the situation for the Macquarie perch there is, well, it's quite dire. This Abercrombie population is genetically unique. It's different from the rest of the fish in the Murray-Darling Basin.
Unique genetics. That raises the stakes even higher.
It really does. And we know what redfin perch can do. They likely wiped out the Macquarie perch entirely in the nearby Lachlan River. Just gone.
Wow. So if the Abercrombie population goes...
We lose that unique genetic lineage forever. It's a serious conservation concern.
So what was done to try and prevent that? I understand there was a refuge strategy.
Yes, exactly.
Yeah.
They tried to create a safe zone. They identified a side tributary, the Retreat River.
Okay.
And the hope was that some waterfalls near the bottom of the Retreat River would act as a natural barrier, stopping the invasive redfin from swimming upstream into the refuge.
Makes sense. A physical barrier.
Right. So between 2010 and 2014, they actually released over 19,000 captive-bred Macquarie perch fingerlings into this Retreat River refuge.
19,000. Wow.
Yeah, big effort. And initial monitoring using traditional methods suggested they were surviving and even breeding up there.
That sounds promising, but it all depends on that barrier holding back the redfin, right?
Absolutely. Which brings us back to the eDNA challenge. Why is it so hard to just, count the DNA and get a population size,
Yeah.
especially in rivers?
Yeah, let's unpack that a bit. What makes rivers so tricky for eDNA quantification?
Well, think about it. Rivers are dynamic. Water flow changes constantly. Sometimes it's fast, sometimes it's slow. That affects how DNA moves, how diluted it gets.
Okay, dilution.
And then there's temperature, water chemistry like pH. These affect how quickly the DNA breaks down, degrades.
Right, so the signal fades at different rates.
Exactly. Plus, the fish themselves. How much DNA they shed depends on their stress levels, their activity, even the time of year. It's incredibly complex.
So trying to get an absolute number, like there are 537 fish in this stretch, is basically impossible with all those variables.
Pretty much, yeah. Especially across larger areas at different times. But Rojahn and the team had a hypothesis. They thought, maybe if we look at a smaller scale, comparing sites close together, or comparing the relative amounts of DNA from two different species at the same site.
Ah, so comparing apples to apples, locally speaking.
Sort of, yeah.
Yeah.
The idea is that maybe those confounding factors, flow, temp, etc., might be similar enough across nearby sites or affect both species similarly at one site that you can get meaningful relative comparisons.
Okay, so not how many redfin are there, but are there relatively more redfin here compared to Macquarie perch or compared to that site downstream?
Precisely. That was a core idea they were testing. Can relative quantitative eDNA data provide useful ecological insights?
So how did they set up the study to test this? The sampling?
They did their eDNA sampling over 4 days back in May 2018. They collected 1-liter water samples from 9 spots.
9 spots. Where were they located?
7 were along the main Abercrombie River itself, covering a range of habitats. And crucially, 2 sites were in the Retreat River Refuge.
Including one above those waterfalls.
Exactly. One site above the falls to see if any redfin DNA had made it past that supposed barrier. And they deliberately sampled near vegetation because that's where redfin perch like to hang out.
Smart. Okay, so they collect the water. What happens in the lab? You mentioned quantitative PCR.
Right, qPCR. It's a step beyond just detecting if the DNA is there. qPCR uses fluorescent probes that light up as the target DNA sequence is copied.
So the brighter the signal.
The more target DNA was in the original sample, it gives you a concentration value. But just getting those raw numbers isn't the end of the story. The analysis needs to be sophisticated too.
Because the DNA isn't evenly spread out in the water, right?
Exactly. It's patchy. You might get a high reading in 1 liter and a low one just a meter away. So they use a statistical approach called a hierarchical Bayesian model.
Okay, that sounds complex. Why Bayesian?
It's particularly good at dealing with that patchiness, that variability you see in environmental samples. It helps estimate the most likely average concentration at a site, accounting for the fact that some samples might randomly have more or less DNA just due to how water swirls around. It gives a more robust estimate of the relative signal strength.
Right, it sort of smooths out the noise to get at the underlying pattern. Did they have controls in place? Lab hygiene is key with eDNA.
Absolutely critical. They use negative equipment controls and blank field controls to check for contamination at every step. They reported only one tiny contamination blip in a lab control, likely from splashover from a high concentration sample nearby, which they accounted for. Good practice.
Okay, so robust lab work, sophisticated analysis. But they didn't rely solely on eDNA, did they? There was conventional monitoring too.
That's right. You need to ground truth. They used electrofishing and also Fike nets. Those are funnel-shaped nets. They did surveys in April 2018, just before the eDNA sampling, and again in May 2019.
The year later, was that second survey planned from the start?
Interestingly, the 2019 survey was partly designed in response to the eDNA results from 2018. They specifically targeted some upstream sites in 2019 because the eDNA had shown something unexpected there.
Ah, so the eDNA guided the later conventional effort. That's neat. Any notes on the conventional methods themselves?
Yeah, they did note that Fike nets seemed to be better at catching the native Macquarie perch compared to electrofishing in this system. Useful observation for future monitoring.
Okay, let's get to the findings. The moment of truth. First up, the Retreat River Refuge. Did the waterfall barrier work? Did they find redfin DNA above the falls?
The good news first? No. They did not detect redfin perch DNA at that critical site above the waterfalls.
Phew. Okay, that's huge.
It really is. It provides strong evidence using this non-invasive method that the natural barrier is effective and the refuge strategy is sound, at least regarding redfin invasion from downstream.
And the Macquarie perch DNA, was it found generally?
Yes, Macquarie perch DNA was detected at all the sites they sampled overall, which confirms they are still present across the system, including in the refuge.
Okay, great news for the refuge. Now, what about the main Abercrombie River, where both fish are present? What did the quantitative comparison show about their relationship?
This is where it gets really interesting. They ran a regression analysis comparing the concentrations of redfin eDNA versus Macquarie perch eDNA at each site in the Abercrombie.
Yeah.
A clear, statistically significant negative relationship. Meaning, at sites where the concentration of the invasive redfin perch eDNA was higher, the concentration of the native Macquarie perch eDNA tended to be lower, and vice versa.
Wow. So the eDNA concentrations mirrored the expected negative impact of the invasive on the native.
Exactly. While eDNA itself can't prove why it doesn't show you a redfin eating a Macquarie perch, the quantitative pattern strongly supports the known ecological dynamic. Higher invader presence correlates with lower native presence. It showed the relative eDNA concentrations were ecologically meaningful.
That's a powerful finding. Okay, now for the big surprise you hinted at. The distribution pattern in the Abercrombie. What was expected versus what the eDNA revealed.
Well, the general expectation, based on how invasions often proceed, was that redfin perch abundance would likely be highest downstream and then gradually decrease as you move further upstream, sort of petering out.
Makes sense. Standard dispersal.
But the eDNA concentrations told a completely different story. Wow. For redfin perch, the eDNA concentrations generally increased from the downstream sites towards the upstream sites.
Increased upstream, that's backwards.
Totally unexpected. It immediately suggested something else was going on. Perhaps a more recent, previously undetected incursion happening in the upper reaches, or maybe a localized population boom up there.
Hmm. But how certain could they be? Could it just be an artifact of eDNA detection being better upstream for some reason?
That's where the follow-up conventional survey in 2019 becomes absolutely crucial.
Yeah.
Remember, they targeted some of those upstream sites specifically because of the eDNA results.
Right, the validation step. So what did the nets and electrofishing find in 2019?
They found high numbers of actual redfin perch fish in those upstream sites by and Gilligan. It confirmed the pattern the eDNA had detected a year earlier.
Wow. So the eDNA really was an early warning system. It picked up on this unusual upstream abundance before it was obvious from traditional surveys.
Precisely. It validated the eDNA approach as a powerful tool for detecting these unexpected shifts in distribution, these potential new fronts of invasion. And interestingly, they also caught Macquarie perch further upstream than previously recorded in that 2019 survey at Bylemans.
Okay, so let's tie this back to the value of quantitative eDNA. What does this study really show it can do for conservation and management?
I think it demonstrates two key things really well. First, that relative eDNA concentrations can help us infer species interactions, like that negative correlation between the invader and the native.
Okay, understanding the dynamic.
And second, maybe even more critically for rapid response, it shows that quantitative eDNA can reveal unexpected distribution patterns, like that upstream redfin hotspot, potentially much faster and more efficiently than traditional methods alone. It's about getting ahead of the curve.
That makes total sense. So for management, this information is gold. Knowing the retreat river barrier is holding means they can focus efforts, right? Like community awareness.
Exactly. The paper points out the biggest remaining threat to the refuge is probably people, accidental introductions, maybe releasing unused bait fish, that sort of thing. So public education in that area is key.
And what about the bigger picture, the fate of that unique Abercrombie genetics?
That remains a serious concern. Even with the refuge, the main river population is under pressure. And we have to remember, captive breeding programs, while essential lifeboats, often lead to a loss of genetic diversity over time. Founder effects, genetic drift. You just don't capture the full spectrum.
So protecting the wild Abercrombie population and its genetics is still paramount.
Absolutely. Future management really needs to focus on maximizing the preservation of that unique genetic heritage. That might mean more genetics research, maybe establishing additional refuge populations if possible, or carefully managing the existing ones to maintain diversity. Losing the Abercrombie population would be a major blow.
So the main takeaway for you, the listener, from this deep dive seems really clear. While getting precise absolute fish counts from eDNA is still, a work in progress.
Still tricky.
Using quantitative eDNA to look at relative patterns, comparing sites, comparing species, gives us incredibly valuable intelligence. It helps understand ecological battles and, crucially, can flag up hidden threats like new invasion fronts much earlier.
It really shifts eDNA from just detection to ecological assessment. And that leads to a final thought, maybe something for you to ponder. If these quantitative eDNA surveys prove consistently reliable and cost-effective as early warning systems, especially in complex rivers all over the world,
Yeah.
what needs to happen next? How do we build the bridges to get this powerful information from the scientists into the hands of managers and policymakers quickly enough to make real-time decisions? That integration into practical conservation frameworks is the next big challenge.
A really important point, moving from research insight to on-the-ground action. Food for thought indeed. Thanks for joining us on The Deep Dive. We'll catch you next time.